So NK cells will express activating inhibitory receptors that will help them in this task. So what's important to note is that on healthy cells, or here what's noted as our target cell, each of our target cells expresses a molecule called MHC class I on its cell surface. So this is a marker that you'll learn about in further detail in other lectures, but it's essentially a marker of self. These are all of our cells pretty much in our body have this MHC class I on the surface, and it tells our immune system that this is my cell. This is a cell that's part of my multicellular organism. So the NK cell, the natural killer cell, has a receptor called the inhibitory receptor, seen here, that recognizes the MHC class I molecule. If the NK cell, or the natural killer cells, sees that there's an MHC class I molecule that it recognizes on a cell that it's approaching, it will know that it doesn't need to kill the cell because it's a self cell, and this leads to inhibitory signaling through the inhibitory receptor that leads to the NK cell not killing this normal cell. Interestingly, if there is a missing or not correct MHC class I molecule that cannot trigger the inhibitory receptor of the NK cell, seen here, then the cell will not receive inhibitory signals that prevent the NK cell from killing that cell. In addition, cells can express NK cell activating ligands that will activate the NK cell to then trigger the killing of the cell via apoptosis. You can imagine that NK cells' ability to induce apoptosis in host cells is an important mechanism by which intracellular pathogens can be eliminated. Intracellular pathogens replicate and exist within cells of the host, and so NK cells are really important in making sure that these cells that are basically factories for the proliferation of intracellular pathogens are eliminated. So this is why NK cells are a really big responder in the innate immune response against viruses and intracellular bacteria. So now we've covered induced innate immune responses to infection. So now let's move on to pattern recognition by cells of the innate immune system. So what do I mean by pattern recognition? So there's different ways of recognizing an infection. So here we're going even deeper from responding to broad classes of, let's say, just bacteria or viruses, as we just talked about with ILCs and NK cells. We're now moving into how do we respond to a very specific type of bacteria or specific type of virus. So you can imagine that an infection can be detected in one of two ways. First, you can either identify the pathogen itself, or you can identify signs of damage to your own body. So the identification of the pathogen is based on the fact that microbes express molecules that are not expressed by mammalian cells, and that microbes can express molecules that we as hosts have, but they're expressed in incorrect places within the cell. And we'll go into what this means. And what's important to remember about the identification of damage is that self-derived host proteins can be induced to indicate cellular infection, damage, or stress. So for example, if there's a nuclear protein that you find in the blood outside of a cell, that is a sign of damage. And we'll go into this in a little bit more detail. So what I want you to remember is the phrase PRRs recognize PAMPs. So what this means is that pattern recognition receptors recognize pathogen-associated molecular patterns, or PAMPs, or they recognize damage-associated molecular patterns, or DAMPs. So PRRs is just a very broad way of describing receptors within our innate immune response that recognize either pathogens or damage-associated molecular patterns. So today we'll be covering toll-like receptors, or Tlrs. We'll be covering nucleotide binding oligomerization domain-like receptors, or NOD-like receptors, or NLRs. And we'll be talking about a few cytoplasmic PRRs that induce what are called type 1 interferons. So let's start off with toll-like receptors. So figure 310 in your textbook has a good summary of the different types of toll-like receptors that are found in our innate immune system, as well as the ligands that they recognize. So this would be a good reference if you're looking for a place to find out what ligands stimulate toll-like receptors. So there's a couple general properties of TLRs that I would like you to remember. So first, here's a schematic of TLRs on a cell surface. So they can sense extracellular space if they're expressed on the cell surface here. So you can see that they're on the plasma membrane, and they're facing out of the cell, able to sense the extracellular environment. They can also sense intracellular space when they're on the membranes of what are called endosomes. So here's the endosome. And here you could see the membrane of the endosome, and the TLRs face the interendosomal space. And in general, some other things to remember is that they are single-pass transmembrane proteins here, and and they have an extracellular region composed of leucine-rich repeats, or LRRs. And they have these cytoplasmic tails that are necessary for downstream signaling. So if you can see here, the cytoplasmic tails of both the cell surface and the endosomal membranes, these tails stick into the cytoplasm, which then leads to signaling pathways. In addition, TLRs, the way that they function is ligand-induced dimerization of two TLR ectodomains, so the outside domains, brings together their cytoplasmic domains here. And these TIR domains of the cytoplasmic domains will interact with TIR domains of other proteins or adapter molecules that leads to further signaling. And the signaling cascade that's induced by the TIR domains of the cytoplasmic domains coming together leads to the production of cytokines and other molecules, leading to a stronger immune response. So now I want to focus in on some example of TLRs. So let's start with the cell surface TLRs. So these recognize, as I mentioned, extracellular pathogens, so pathogens that are outside of the cell. And these include a variety of different kinds of TLRs. So for example, we have TLRs 1, 2, and 6. So TLRs 1, 2, and 6 recognize various components of gram-negative bacterial cell walls, such as diacylipopeptides and triacylipopeptides. And they're expressed on macrophages, DCs, eosinophils, and mast cells. And it's pictured here that they're in heterodimers, which is unique because ligand binding induces either heterotetramers of either TLR2 with TLR6 or TLR1. So most of the other TLRs function as homodimers, so two TLRs domains of the same TLR, but TLR1, 2, and 6 act interestingly because they are heterodimers. Next, we have TLR5, which recognizes monomeric flagellin of bacteria, and it's expressed on macrophages, dendritic cells, and intestinal epithelial cells. And we have TLR4, perhaps one of the most famous TLRs, and it recognizes bacterial lipopolysaccharide. And it works with a variety of additional host accessory proteins called MD2 and CD14, and it's expressed on macrophages and DCs. So those are the major cell surface TLRs that you'll be hearing about throughout the rest of your lectures. Now let's move on to the endosomal TLRs. So these recognize nucleic acids, and that might be somewhat surprising because you could think, well, hosts have nucleic acids, hosts have DNA, hosts have RNA, but you'll see in a few slides why this is important. So first, let's start out with TLR3. So TLR3 recognizes double-stranded RNA, and it's expressed on macrophages, conventional DCs, and intestinal epithelial cells. And double-stranded RNA is something that host species typically don't have, so TLR3 recognizes a nucleic acid species that mammalian cells typically do not have. In addition, we have TLR7 and 8. So both TLR7 and 8 recognize single-stranded RNA. So typically in healthy cells, single-stranded RNA is confined to the nucleus and to the cytoplasm where translation occurs, and not to the endosome specifically. So this would signal that even though host cells have single-stranded RNA, the mislocation or the incorrect location of single-stranded RNA signals to the cell that there could be an infection. TLR7 is expressed on a subset of dendritic cells, B cells, and eosinophils. And TLR8 is expressed on monocytes and macrophages. However, the exact role of TLR8 is not really well known. You can see that they recognize the same nucleic acid species, so that's still an area of ongoing research. Next, let's move on to TLR9. So TLR9 recognizes unmethylated CpG dinucleotides. So typically in mammals, CpG dinucleotides are methylated, and they're unmethylated in bacteria or viruses. So this sort of non-traditional marking of nucleic acids allows our immune system to distinguish between dinucleotides that originate from host cells or from invading pathogens. And TLR9 is expressed by, again, a subset of DCs, B cells, and eosinophils. So TLR activation induces the expression of a variety of different inflammatory cytokines that allow for the continuation of an immune response. So as I mentioned previously, TLR molecules dimerize and then recruit other signaling pathways based on their Tlr domains that are in the cytoplasm, as seen here. So for example, in this situation, the TLR has recruited two different molecules called IRAK1 and IRAK4, which then trigger the activation of the E3 ubiquitin ligase, TRAF6. This protein, TRAF6, is then polyubiquitinated, creating what's called a scaffold for the activation of TAC1. And then TAC1 associates with IKK and phosphorylates IKK beta, which then leads to the phosphorylation of I-kappa B. And I-kappa B is interesting because it holds and keeps inactive a protein called NF-kappa B. So NF-kappa B is a really critical transcription factor that is important in initiating the transcription of cytokine genes. So I know I went through this cascade very, very quickly. Let's go through it again, and I'll point out what I want you to remember. First, the TLRs, upon binding to their ligand in the extracellular or endosomal space, will dimerize. This brings together their cytoplasmic tails, as I mentioned before, seen here. This dimerization and bringing together of the two Tlr domains in the cytoplasm recruits other proteins to the dimerized cytoplasmic domain, seen here. And this leads to a signaling cascade that then releases a cytokine gene transcription factor. Now, these in-between steps will be gone through in further lectures. However, what's important to note, and I want you to recognize and remember for a couple slides from now, this protein MyD88, which binds to the Tlr domains of our TLRs. TLR activation can induce another type or a specific type of cytokine family called type 1 interferons, which I'll go into in a few slides. But what I want you to take home from this slide, which is figure 316 from your textbook, is that endosomal TLRs, so TLR3 and TLR7, shown here, go through similar signaling cascades as the cell surface TLR in the previous slide that lead to the activation of transcription factors that lead to what are called type 1 interferon genes. So these are antiviral genes that we'll go through in a few slides. But so what's critical to remember, and what I want you to take away from here, is again you see this protein MyD88 here, and you see a new protein TRIF here. So these proteins that I've pointed out, TRIF and MyD88, are different adapters that TLRs use in order to signal. So these are adapters that'll bind to those dimerized Tlr domains in the cytoplasm upon dimerization of the Tlrs that lead to downstream signaling that leads to the expression of cytokines. So you can see that MyD88 is a very popular adapter that's used by TLRs. We also have TRIF and TRAM and MAL. But what's important to remember in terms of potentially an experimental context or a clinical context, if you were to knock out just one or eliminate the function of one TLR, this renders the body or the body of the host susceptible to infection by whatever this molecule that it's recognizing. So for example, if you were to knock out TLR4, which recognizes bacterial LPS, the host or the experimental animal will be more likely to be infected by a bacteria. However, if you were to knock out or eliminate a TLR adapter, such as MyD88, the host or the patient is then more susceptible to pathogens that are that express everything that is recognized by TLRs that use that specific adapter. So for example, if you have a patient or an experimental animal that is lacking MyD88 or lacking a functional MyD88, this experimental animal will then be susceptible to anything that's recognized, any pathogen that has components that are recognized by TLR2, by TLR4, 5, you can see. So that's something that's really important to keep in mind about TLR signaling. Next, let's move on to our Nlrs, or our NOD-like receptors. Nlrs recognize bacterial cell wall peptidoglycans, and this is a really, really large group of cytoplasmic innate sensors, and some initiate the same inflammatory responses as TLRs, but others trigger distinct pathways that lead to cell death and the production of other pro-inflammatory cytokines. So what the general properties of these are is that NOD proteins, or Nlrs, reside within the cytoplasm in an inactive form. And then when bacterial ligands are found within the cytoplasm where these Nlrs are expressed, this leads to the dimerization of NOD proteins, which then leads to the recruitment of the adapter RIP2, which then activates TAC1. So this is a very similar cell signaling that you saw from TLRs, because NF-kappa B, again, this is a cytokine transcription factor, is induced by the Nlrs in this case. But as I just mentioned, Nlrs also have the unique property of being able to induce cell death in the host cell that's occurring. So again, the induction of apoptosis, or cell death, in host cells that contain intracellular bacteria or viruses is really critical in minimizing the locations where these pathogens can replicate and produce more of themselves. So here what happens is that NLRP3, or which is a component of the subfamily member of NOD-like receptors in the NLRP3 family. So here NLRP3 is activated, which leads to the formation of NLRP3 oligomers, which leads to the proteolytic cleavage of procaspase-1, which releases not only pro-inflammatory cytokines, but also leads to induction of cell death. So again, NOD-like receptors compared to TLRs, although they have similar functions in triggering the transcription of cytokines and other pro-inflammatory proteins, Nlrs can also trigger the cell death of the host cell by triggering the proteolytic cleavage of procaspase-1. Now let's jump into the term that I've been talking about a little bit and referenced a few times, called type 1 interferons. So before we talk about what type 1 interferons are, let's very, very briefly talk about viruses, in that viruses exist in extracellular forms, but then invade the host cell and produce a variety of replication intermediates here, and then they assemble and then bud from the host cell. And interferons are signaling molecules that are induced by virus infection that stimulates the transcription of genes that act in every single component of this pathway. So whether it's external virus, the entry of the virus into the cell, various forms of replication, the formation of mRNA, translation and exit, interferons and type 1 interferons act again on every single portion of this to minimize the production of more viruses, and we'll be going into this in more detail in future lectures. So one big family that produces type 1 interferons are called RIG-I-like receptors, which detect cytoplasmic viral RNAs. So here what happens is that there's cytoplasmic replication of a virus, seen here. And this cytoplasmic replication of the virus leads to uncapped RNA with a 3' triphosphate. Now, mammalian mRNA is capped and does not have a 3' triphosphate, so this RNA is recognized by RIG-I, seen here. And the viral RNA alters the conformation of RIG-I and induces binding and aggregation with another protein called MAVs, which is found on the surface of mitochondria. And then there is polyubiquitination that occurs, and then this aggregated MAVs protein then induces the release of NF-kappa B, as we've previously seen. So what's important to remember here is that RIG-I is sensing a type of nucleic acid that's formed by viruses that mammalian cells do not have, leading to the induction of inflammatory cytokines and also type 1 interferons. We also have a protein called STING that is able to sense double-stranded DNA. So what happens is double-stranded DNA should not be found in the cytoplasm, typically in a mammalian host cell. But if you have a double-stranded DNA virus, this double-stranded DNA activates a protein called cGAS, and cGAS will then produce cyclic GAMP from ATP and GTP. And this cyclic GAMP will bind to the STING dimer that's in the ER membrane and activate its signaling. And this activation of signaling stimulates the activation of IRF3, which is the main transcription factor for type 1 interferons. So again, type 1 interferons are really critical in establishing what's called the antiviral state. And Tlrs and Nlrs are capable of triggering the induction of type 1 interferons, but the main players in this are cytoplasmic viral sensors, such as RIG-I. So now that we've gone through a very general idea of what pattern recognition is, I want to go back to this concept of pattern recognition receptors recognize pathogen-associated molecular patterns or damage-associated molecular patterns. So this is a really critical aspect of our innate immune system. It allows us to recognize specifically what kind of pathogen is invading our bodies to then tailor the response such that we're not triggering an antiviral response against a helminth infection, for example. So this is a really critical concept that will come up and be reiterated in future lectures. So we've gone through now a very broad overview of the innate immune response, starting with the importance of anatomical barriers and their initial chemical defenses. Specifically, epithelial surfaces are our body's site of interaction with the external world, and it keeps our internal milieu safe. However, if these barriers are, if pathogens go through these epithelial surfaces, we have additional defenses, such as the antimicrobial peptides and proteins that our epithelial cells and phagocytes produce. If the infection is not prevented at this stage, non-specific effector cells such as phagocytes and neutrophils are really critical in launching a very early response. And then from this initial response, there's the induction of the inflammatory response. And if these initial effector cells are unable to clear the infection, then we have pattern recognition of the pathogens that are coming in by our Tlrs Nlrs, or cytoplasmic viral sensors. And we also have induced innate responses to infection, such as the chemokines and cytokines that are produced, and the function of ILCs and NK cells in the infection. So again, I want to reiterate the idea that throughout the course of an immune response, we go from being able to target a large variety of pathogens to a very specific pathogen, such that the pathogen specificity goes from very narrow to highly, highly specific. So today we covered innate immunity and its roles in the very early responses against pathogens. And the next portion of the immune response, if the innate immune response is unable to clear the infection, is the adaptive immune response. So in the next lecture, we're going to be talking about very broadly what the adaptive immune response is and its unique properties relative to the innate immune response, as well as the bridge between the innate and adaptive immune system. So yeah, please stay tuned for that lecture.